Greetings! Welcome to my channel, Human, Evidently, where I provide evidence-based videos on human origins, paleoanthropology, archaeology, and evolutionary science for adults. My presentation videos are developed from the study material I build for my undergraduate courses and scholarly sources, and can be used to supplement your own learning in high school or college-level classes. Today's lecture examines fossil hominin identification through comparative morphology. The goal is to move beyond memorized species labels and toward anatomical reasoning. Paleoanthropologists often work with incomplete, fragmentary, distorted, or regionally variable specimens, so identification depends on the patterning of preserved traits across the cranium, dentition, pelvis, foot, and postcranial skeleton. A fossil specimen should be read as a character profile: cranial vault shape, canine form, premolar morphology, brow ridge structure, pelvic organization, haliacal position, and long bone anatomy each contribute different kinds of evidence. Some traits are highly informative. Others are useful only when interpreted in combination with stronger indicators. Brain size, for example, is often tempting as a quick identifier, yet early hominins can be small-brained, and several later archaic humans can overlap in cranial capacity. Prognathism presents a similar problem. It is present in non-hominin apes and in many early hominins. A thick brow ridge may occur in multiple taxa within Homo. A careful identification, therefore, requires trait clusters rather than isolated features. The central question for each specimen is, which anatomical pattern best accounts for the preserved evidence? Identification logic. The first step in fossil identification is description. Before assigning a taxon, the visible anatomy should be determined and understood in its context. Are we looking at a cranium, maxilla, mandible, femur, tibia, ulna, pelvis, foot? And how was it found? Is this a reconstructed specimen or one fully preserved in articulation? Do we have all of it or just this one component? The preserved anatomical regions determine which questions you can answer. A maxilla may preserve canine size, diastema, and dental arcade information. A pelvis may provide evidence relevant to bipedal mechanics. A femur may help you understand hip and knee morphology, even if all you have is the femur itself. A cranial vault may show you information about brain case shape, brow ridge form, and vault thickness, depending on the quality of the specimen. The second step is ranking traits by evidentiary strength. Strong early hominin indicators include an anteriorly positioned foramen magnum, reduced canines, and reduction or absence of the CP3 Honing Complex. These traits are particularly useful in distinguishing early hominins from non-hominin apes. Weaker standalone indicators include brain size, facial prognathism, and brow ridge size. These features remain useful, but require support from other features to have a determinate value on what taxon that specimen belongs to. The third step is determining the taxonomic level supported by the evidence. It's important to remember that the animal kingdom is a nested hierarchy, which means that your fossils will have a lot in common. Some features are unique to certain taxa, though, and these will help you separate your specimens into more and more specific identification labels. The fourth step is to resist overinterpretation. A fossil can preserve a mosaic of primitive and derived traits. Mosaic anatomy should be treated as expected evidence in hominin evolution, especially in early taxa such as Ardipithecus ramidus. Sometimes, you can only get as far as understanding which genus your specimen fits within, but not the actual species name. Morphology is not the only way to determine what species a specimen belongs to, and we'll cover more in-depth problem solving in that domain another time. For now, let's move on to an important identifier that helps scientists distinguish non-hominin apes from hominin apes: the CP3 Honing Complex. The CP3 Honing Complex is one of the most useful dental systems for distinguishing many non-hominin apes from early hominins. In this system, the upper canine is large, projecting, and sharpened against the lower third premolar. The upper jaw may preserve a diastema to accommodate the opposing canine. The lower third premolar is often sectorial, elongated, blade-like, and dominated by a single main cusp. Hominins show reduction or loss of this functional complex. The canines become shorter and less projecting. Diastemata are reduced or absent. The lower third premolar becomes less sectorial and more molarized through time. This dental transition is central to early hominin identification. A specimen with a large projecting canine, a clear diastema, and a sectorial lower third premolar strongly supports a non-hominin ape identification. A specimen with reduced canines and no functional honing complex is more consistent with hominin status. Facial projection and cranial capacity should not override this dental evidence. Early hominins may be prognathic and small-brained, so those traits cannot serve as decisive exclusions. For practical identification, the key question is whether the canine and lower third premolar form a functional sharpening system. If that system is present, non-hominin ape status is strongly favored. If that system is reduced or absent, hominin status becomes more plausible, especially when other traits support the same conclusion. Homo heidelbergensis. Homo heidelbergensis occupies an important position in Middle Pleistocene human evolution. It is generally associated with a large brain, thick cranial bones, a receding frontal bone, and a strong, continuous supraorbital torus. The cranial vault is often long and low, retaining an archaic form compared with later Homo sapiens. Reitmeyer, 1998, treated Homo heidelbergensis as central to discussions of Middle Pleistocene human evolution, particularly in relation to later Neanderthal and modern human lineages. Some students struggle to differentiate heidelbergensis crania from neanderthalensis crania. Some Middle Pleistocene specimens attributed to heidelbergensis may show posterior occipital expansion or an occipital bun-like projection, a trait commonly associated with Neanderthals. Heidelbergensis has a distinct craniofacial pattern, including the absence of a Neanderthal-like retromolar space, the thickness of the brow ridge, the overall vault shape, and the degree of facial specialization. The most defensible identification of heidelbergensis relies on a cluster of traits: large cranial capacity, archaic cranial vault form, thick continuous supraorbital torus, receding frontal bone, thick cranial bones, and no retromolar space. Individual traits may overlap with other archaic humans, so it's important to get as complete a read on the traits as you can. Paranthropus aethiopicus. Paranthropus aethiopicus belongs to the robust Australopith lineage and is most clearly recognized through its masticatory anatomy. The post-canine teeth are enlarged relative to the anterior dentition. The zygomatics are positioned anteriorly, the sagittal crest is present, and the temporal fossae are large. These traits indicate a craniofacial system organized around powerful chewing musculature. KNM-WT17000, often called the black skull, remains the classic specimen for this taxon. Walker et al., 1986, described it as a 2.5 million-year-old robust Australopith from west of Lake Turkana. Its morphology combines robust masticatory traits with a more primitive cranial form. Compared with later Paranthropus boisei, Aethiopicus shows stronger prognathism and a more ape-like cranial base. Wood and Constantino, 2007, emphasize that robust Australopith diversity should be interpreted through species-specific combinations rather than through a single generalized robust template. To summarize, Aethiopicus's strongest signals are large postcanines, anteriorly placed or flared zygomatics, sagittal cresting, and large temporal fossae. These traits work together as a masticatory complex. Brain size and prognathism may help with species-level comparison, but the robust chewing apparatus carries the primary identification. Homo erectus, and OH9. OH9, or Olduvai hominid 9, is a significant Homo erectus calvaria from Tanzania. Leakey, 1960, described the specimen from Olduvai Gorge, and later work has emphasized its unusual combination of African geography and cranial morphology, resembling traits often associated with Asian H. erectus. Antong, 2003, characterized erectus as a geographically and temporally variable species, making OH9 especially useful for examining variation within the taxon. The specimen has a low and receding frontal bone, a supratoral sulcus, a massive supraorbital torus, a long and low cranial vault, and an angled occipital region. Its brow ridge is exceptionally thick. Its cranial capacity is larger than that of many earlier East African Homo specimens. Its cranial vault morphology aligns with the broader erectus pattern, especially the long, low vault and robust cranial architecture. The most careful interpretation is that traits often associated with later East Asian Homo erectus were also present within broader African erectus variation. OH9 should not be used as evidence for early Homo sapiens, Neanderthal-like status, fossil distortion as a primary explanation, or direct replacement of African populations by East Asian erectus. Its value lies in demonstrating that regional morphology does not always map neatly onto geography. Australopithecus, cranial and dental traits. Australopithecus includes small-brained, prognathic, bipedal hominins, with substantial variation among species. Common traits include a small cranial capacity, a sloping frontal bone, facial prognathism, and postcranial evidence for bipedality. These features establish broad, genus-level expectations, but species-level identification requires more precise comparison. Sagittal cresting may appear in some Australopith individuals or species, yet it does not define Australopithecus as a whole. In Australopithecus afarensis, sagittal or compound cresting has been interpreted as variable and likely associated with size and sexual dimorphism in some individuals. Kimball, Rack, and Johansson, 2004, emphasized the importance of cranial variation within A. afarensis, including traits relevant to masticatory anatomy and cranial form. Haile Selassie et al., 2019, in their description of a 3.8 million-year-old cranium from Warranso Mill, also contributed to broader discussion of Australopith cranial variation. Sagittal crests must be distinguished from sagittal keels. A sagittal crest is a raised midline ridge associated with temporalis muscle attachment. A sagittal keel is a lower, rounded midline thickening of the cranial vault. Balzo, 2013, analyzed cranial vault thickening and parasagittal keeling in Homo erectus, showing that keel-like morphology belongs to a different anatomical and interpretive category than robust Australopith cresting. In identifying Australopiths, the better approach is to use cresting as one feature within a broader pattern. A small brain and prognathic face may suggest an early hominin, while masticatory robusticity, zygomatic position, and postcanine size help distinguish gracile Australopiths from robust Australopiths. Ardipithecus ramidus, Mosaic anatomy. Ardipithecus ramidus lived about 4.4 million years ago at Aramis in the Middle Awash region of Ethiopia. White et al. Described Ardipithecus ramidus as a taxon that reshaped interpretations of early hominin evolution. Its anatomy combines reduced canines, a small brain, a grasping hallux, and pelvic traits consistent with some bipedal capacity. This combination is best understood as mosaic anatomy. Different anatomical systems preserve different evolutionary signals. The dentition supports hominin affinity through canine reduction. The cranium preserves a small brain and prognathic face, traits that remain primitive relative to later hominins. The foot retains a divergent hallux, indicating grasping ability and arboreal competence. The pelvis shows reorganization relevant to upright walking. White et al. Argued that Ardipithecus challenges models that treat the human-chimpanzee last common ancestor as essentially chimpanzee-like. The anatomy of Ardipithecus ramidus suggests a form of locomotion that included terrestrial bipedality while retaining climbing ability. This pattern does not align with modern human obligate bipedality or with chimpanzee-like knuckle walking. Ardipithecus ramidus is an excellent example of early hominin mosaic evolution. It shows us that hominin status does not require obligate or even habitual bipedality, and early hominins may preserve derived dental and pelvic traits alongside primitive cranial and pedal traits. Ardipithecus ramidus, cranial traits. The cranial traits of Ardipithecus ramidus include an anteriorly positioned foramen magnum, facial prognathism, small brain size, short upper canines, and non-triangular upper canine form. Sua et al. Described the A. ramidus skull and emphasized its implications for hominid origins. Kimball et al. Further examined the cranial base, arguing that A. ramidus shares relevant basicranial features with later hominins. The foramen magnum is frequently discussed in relation to posture. In habitual bipeds, the foramen magnum tends to be positioned more anteriorly under the cranial base, helping align the head over the vertebral column. In A. ramidus, this trait provides partial support for hominin affinity and altered posture. Cranial evidence alone remains limited for reconstructing locomotor behavior. The postcranial skeleton provides the stronger evidence for how the animal moved. Reduced canine morphology is also important. The upper canine is short and does not preserve the projecting triangular form associated with a functional honing complex. This dental reduction supports early hominin status. When the cranial base and canine evidence are interpreted together, A. ramidus fits within early hominin evolution, even though its brain size and facial projection remain quite primitive. Ardipithecus ramidus, foot and pelvis. The locomotor anatomy of Ardipithecus ramidus supports facultative bipedality with retained arboreal competence. Lovejoy, Latimer, Sua, Asfaw, and White, 2009, interpreted the pelvis and femur as evidence for the emergence of upright walking. The pelvis appears reorganized in ways relevant to upright posture and bipedal movement. These traits indicate that Ardipithecus ramidus was capable of terrestrial bipedality. The foot preserves a divergent opposable hallux. This trait indicates grasping ability and continued use of arboreal substrates. A divergent hallux limits the formation of the rigid propulsive lever seen in later Australopiths and humans. The foot therefore argues against fully habitual terrestrial bipedality. Lovejoy, Simpson, White, Asfaw, and Sua, 2009, characterized Ardipithecus as a careful climber rather than a knuckle walker. This interpretation places Ardipithecus in a locomotor category distinct from both modern African apes and later habitual or obligate bipeds. The strongest classification is facultative terrestrial bipedality, with substantial retained climbing adaptations. AL288-1, Lucy, and long bone identification. AL288-1, commonly known as Lucy, is attributed to Australopithecus afarensis. Long bones from this specimen provide an opportunity to practice postcranial identification and locomotor inference. The method begins with anatomical orientation: proximal and distal ends, articular surfaces, shaft shape, and the relationship between bone form and function. The femur is identified by its spherical head, distinct neck, shaft, and distal condyles. In bipedal hominins, femoral morphology is especially informative due to the relationship among the hip, knee, and body weight transmission. The tibia can be recognized through its broad proximal plateau and robust shaft. It is central to weight-bearing in the lower limb. The ulna can be identified through the olecranon and trochlear notch region at the proximal end, which form part of the elbow joint. Long bone identification should not rely on overall length alone. Articular surfaces and functional anatomy provide stronger evidence. In fossil hominin studies, these bones can also support behavioral inference, especially when they preserve adaptations related to bipedal gait. Comparative trait profiles. A non-hominin ape identification is strongly supported by large projecting canines, a diastema, and a retained C/P3 Honing Complex. These traits indicate a functional dental system that differs from the reduced canine pattern of early hominins. Ardipithecus ramidus is characterized by reduced canines, small brain size, an opposable hallux, and pelvic traits consistent with some bipedal capacity. Its anatomy supports early hominin status while preserving substantial arboreal adaptations. Australopithecus generally combines small brain size, facial prognathism, a sloping frontal bone, and bipedal adaptations. Species-level identification requires more precise comparison, particularly in cranial and dental details. Paranthropus is distinguished by robust masticatory anatomy, large postcanines, anteriorly positioned or flared zygomatics, sagittal cresting, and large temporal fossae. These traits indicate a specialized chewing apparatus. Homo erectus, including OH9, is associated with a long, low cranial vault, thick supraorbital torus, supratoral sulcus, robust vault bones, and angled occipital morphology. Its variation across regions should be interpreted cautiously. Homo heidelbergensis is identified by large brain size, thick cranial bones, a strong continuous brow ridge, archaic cranial form, and absence of a retromolar space. This profile supports a Middle Pleistocene Homo identification. Practice identification, hominin or non-hominin ape. Let's practice identifying specimens by their traits. Here you have a specimen preserving a large projecting canine, a diastema, and a sectorial lower third premolar. I will give you 30 seconds to decide on your answer. You are correct if you determine that this specimen should be identified as a non-hominin ape. The key evidence is the CP3 Honing Complex. If studying for an exam, keep in mind that the strongest answer would ideally provide both the identification and the supporting trait. A response such as, "This is a non-hominin ape," as supported by the presence of a CP3 Honing Complex, is appropriate, but more detail about the dental arcade of the specimen would likely yield a more favorable grading response. Practice identification, Homo heidelbergensis. Now, you have a specimen with a cranial capacity around 1,250 cubic centimeters, a long, low cranial vault, some postorbital constriction, a receding frontal bone, a thick supraorbital torus, some facial prognathism, parietal expansion, occipital projection, thick cranial bones, and no retromolar space. You can and should use the image on the slide to help you if needed. I will give you 30 seconds to decide on your answer. All right, let's walk through it together. The traits described support a classification of Homo heidelbergensis. But why? Well, the large brain and archaic cranial form place the specimen within the Middle Pleistocene, and likely within Homo. The thick brow ridge and long, low vault distinguish it from modern Homo sapiens. The absence of a retromolar space argues against a Neanderthal identification. What other species could it be? A useful practice would be to try and argue for any other species identification based off those traits. How many species share the listed traits with Homo heidelbergensis? If you find any, then what methods would you use to decide officially which species your specimen belongs to when significant traits overlap? Paleoanthropology is all about being able to defend your own ideas. So share your thoughts in the comments and discuss with other commenters. Practice identification. Robust Australopith. On this slide, you'll see a term bank made up of colorful stars. The stars list traits used to distinguish robust Australopithecines from each other: large postcanine teeth, anteriorly flared zygomatics, sagittal cresting, large temporal foci, and high postorbital constriction. Sort these terms into the three categories to the left. The categories are robust masticatory anatomy, facial form, and neurocranial pattern. I'll leave you 50 seconds to decide on your answers. So how do you know the difference between the species within the Paranthropus genus? When comparing Paranthropus aethiopicus and Paranthropus boisei, stronger prognathism, smaller brain size, and a more primitive cranial form fit aethiopicus better. Boisei shows a more derived, robust pattern, especially in its extreme facial and postcanine specialization. And, despite its name, Paranthropus robustus is less robust than Boisei. The East African, Boisei, has a more massive skull, larger molars, and a deeper jaw, compared to the relatively smaller, more gracile features of the South African robustus. As you can see, identification should focus on the full craniofacial profile and location, rather than robusticity alone. Practice interpretation, OH9. Members of the same species showed variation based on things like region, sexual dimorphism, and age. Given that logic, why does OH9 display Asian-like Homo erectus traits despite being found in Africa? I'll give you 30 seconds to decide on your answer before proceeding with the answer. OH9's African locality and East Asian-like cranial morphology complicate simple regional categories. At present, the most parsimonious explanation is that these traits were not restricted to Asian populations, but occurred within the wider morphological range of Homo erectus by approximately 1.4 million years ago. The fossil's massive supraorbital torus, angular torus, frontal keel, mid-sagittal thickening, high cranial capacity, and long low vault all fit within the broader Homo erectus pattern. These, combined with the age and location of the fossil, support the most parsimonious solution as being intraspecific variation. Practice classification, sagittal crests and sagittal keels. When identifying fossils based off crania, it is useful to know the difference between sagittal crests and sagittal keels, and to know, of the species who have them, which always have them versus which only sometimes have them. Pause here and determine your answers before proceeding. You have 5 seconds to pause the video before the answers will play. True sagittal crests are best documented in Paranthropus aethiopicus and Paranthropus boisei. Paranthropus robustus shows sagittal cresting most clearly as a sexually dimorphic trait, especially in larger presumed males. Some larger, presumed male Australopithecus afarensis specimens may have sagittal cresting, though this is not a defining feature of the species. Australopithecus garhi and possibly Australopithecus africanus belong in the limited documentation category. STW573, Littlefoot, is a subject of taxonomic dispute, and I strongly suggest taking time during or after completing this video to read about the unique circumstances surrounding this mesmerizing specimen. Homo erectus displays a sagittal keel, not a sagittal crest. Homo heidelbergensis and other Middle Pleistocene Homo specimens may show keel-like thickening in some cases. Fun fact, a small percentage of modern humans displays sagittal keeling. This is another example of intraspecific variation. Practice interpretation. Ardipithecus ramidus as a facultative biped. The findings surrounding Ardipithecus ramidus challenged long-held evolutionary theories, specifically that human ancestors resembled modern chimpanzees. The 15-year study of this extremely fragile early hominid sparked significant scientific debate regarding her locomotor habits, combining walking and climbing, and her role as a direct human ancestor. So now's your chance to weigh in on the debate. What traits suggest Ardamidus was a facultative biped? I will give you 40 seconds to decide on your answer. A. ramidus is best classified as a facultative biped with retained arboreal adaptations. Evidence for bipedality comes from the pelvis and femur, which Lovejoy et al. Interpreted as showing changes relevant to upright walking. Evidence against habitual terrestrial bipedality comes from the divergent hallux, which indicates grasping ability and retained climbing behavior. If you're studying for an exam, a solid answer will separate evidence for bipedality from evidence against full terrestrial commitment. The pelvis supports upright walking capacity. The foot preserves arboreal function. The combined evidence supports a locomotor mosaic rather than a habitual bipedal pattern. Practice identification, long bones. You'll need to learn how to identify bones through articular surfaces, proximal and distal anatomy, and functional orientation. Overall shape may help, but diagnostic ends provide stronger evidence. I will give you 30 seconds on each slide to determine which image is which long bone. While you make your decisions, here are some clues. A humerus can be identified by its rounded proximal head and broad distal end. A femur can be identified by its spherical femoral head on a neck and distal condyles. The femoral head and neck are especially distinctive, and the distal condyles articulate with the tibia at the knee. An ulna can be identified by the olecranon and trochlear notch region at the proximal end. A tibia can be identified by its broad proximal plateau and robust shaft. These features reflect functional anatomy. The ulna contributes to the elbow joint, while the tibia is a major weight-bearing element of the lower limb. Long answer synthesis. These long-form questions can really get your brain working to help connect the ideas we covered in this lecture today. You don't have to answer them now, but consider reading more from the sources in the description and at the end of this slideshow to help you cement your growing fossil identification foundation. Maybe throw some of your answers in the comments and challenge each other in some healthy discussion. Fossil hominin identification by morphology is a comparative process that requires you to know the different anatomical traits of the species you may come across in the field. A reliable morphological interpretation weighs dental, cranial, pelvic, locomotive, and postcranial evidence together. Which interpretation accounts for the greatest amount of preserved anatomy with the fewest unsupported assumptions? That question keeps fossil identification tied to evidence, and therefore open to reclassification or reinterpretation when new evidence is found. Special thanks to my Patreon subscribers: Red Fox the Starfish, Nori the Salty Cat, and Gary, Jack, and Mark the Porpoises. The value of your kind support is immeasurable. Thanks for spending this time with me on Human, Evidently. I hope you're leaving with a clearer understanding of the subject and a deeper curiosity about the story of life, evolution, and our place within it. Until next time, keep asking those good questions. If you want to support my channel, please consider subscribing here and on Patreon, where you can access my content for free or contribute a small donation to get your name at the end of my videos. I'm working on a series of educational material for middle grade readers and 2 science fiction novels. Such endeavors are only made possible by contributions from viewers like you.